Hello, thank you for the invitation. It's my pleasure to be here today. I'm going to talk about recent advances in cannabinoid, in phytocannabinoid and endocannabinoid research. I'm a scientist. I work with Professor Reem Smoum-Jaouni. So this is the recent work that has been done in our lab in collaboration with other labs. Over the last few decades, research on cannabinoids has gone through several distinct phases. Phytocannabinoids, endocannabinoids, and endocannabinoid-like research. And thousands of publications have been published on cannabinoids, and some of them are already in use as therapeutic agents. Phytocannabinoids research, I'm going to cover nausea, anxiety, and depression. Cannabis sativa, it contains many bioactive compounds, and the most representatives are cannabinoids, flavonoids, and terpenes. And among the cannabinoids, we have the tetrahydrocannabinol, THC, which is responsible for the cannabis psychoactive effects. And other main cannabinoids include the tetrahydrocannabinolic acid, cannabinol, cannabidiol, and cannabidiolic acid. Now, cannabinoids, they are biosynthesized in their acid form in the plant tissue. And cannabidiolic acid is the main biosynthetic precursor of the acids of CBD, THC, and cannabichromene. And these can generate their decarboxylated counterparts under the action of heat or light. CBDA will give CBD, THCA will give THC, cannabichromenic acid will give cannabichromene, and cannabidiolic acid will give cannabidiol. Now, cannabidiolic acid, which is the major constituent of cannabis sativa, was first isolated in 1955, and its structure was elucidated in 1965. It has been shown to inhibit breast cancer cell migration and to have anti-nausea and anti-anxiety effects. Now, the instability of this compound weakens the case of developing it as a medicine, and therefore we decided to compare it with its methyl ester. Since the Since decarboxylation of the acids, it proceeds through the formation of the carboxyl anion, and this is not readily formed in the methyl ester. And indeed, we found that the CBD methyl ester is stable under different conditions up to 6 months. Now, in collaboration with Professor Roger Partwee and Linda Parker, and knowing that the endocannabinoid system plays a major role in the regulation of nausea and anxiety, we were able to see that the CBD methyl ester was much more potent than the CBDA in enhancing the serotonin 1A receptor activation, and therefore it was much more effective in reducing the acute nausea and anticipatory nausea induced behavior in the rat gapping model. Also, CBDA methyl ester proved to be an anti-anxiety compound in a model of stress-induced anxiogenic responding. We were able to see that when the rats, they received a kind of stress, it is the food shock stress, and 24 hours later they were given vehicle or CBDA methyl ester, and then they were placed in the dark compartment of the light dark box, and the time that is spent in the light box, it was tracked. Now, if the mice spend time in the light box, then they are not anxious. But if they spend time in the dark box, then they are anxious. And indeed, we found that the food shock greatly enhanced the stress-induced behavior responding in the food shock, stressed mice, and CBDA methyl ester, it reduced this effect, meaning that the CBDA methyl ester has anti-anxiety effects on the mice. Now, in order to expand the results that we got with the CBDA methyl ester, that it has anti-anxiety effect, and knowing that the serotonin 1A receptor is important for the treatment of human depression, we were able, together with Professor Aaron Veller, to see that CBDA methyl ester, it reduces the antidepressant behavior in rats, and usually the antidepressive-like behavior, the depressive-like behavior in mice are seen as increased immobility and decreased swimming in the forced swim test, and CBDA methyl ester decreased the immobility and increased the swimming in these rats. Now, the endocannabinoids and endocannabinoid-like compounds research, I'm going to cover here two types of topics, which are antimicrobial potential and addiction. Now, after the discovery of THC in 1964, the first endogenous lipophilic molecule that was able to activate the CB1 and the CB2 that are activated by THC was discovered. And this compound was identified in 1992 to be anandamide. And shortly after, another compound, which is important endocannabinoid compound, was discovered, which is 2-arachidonyl glycerol, 2-AG. Following that, other omega-6 fatty acid compounds that have cannabimimetic properties as well, such as nolladine ether, virodamide, and N-arachidonyl dopamine, NADA, they were listed among the endocannabinoid family. And in the last 25 years, anandamide and all of these compounds, they had key biological activities in the central nervous system and in the periphery. Now, compounds that are considered amides of long-chain fatty acids with amino acids or with ethanol amines, they represent a major group of endogenous lipids. And these compounds, they are found in the brain and in the part of them in the periphery. They do not bind to the cannabinoid receptors, CB1 or CB2. However, they have a cannabinoid-dependent activity. And therefore, they are considered as parts of the endocannabinoid family. And these compounds, they have numerous physiological functions in the mammalian tissues. For example, palmitoyl-ethanolamide, PEA, it is anti-inflammatory, anti-nociceptive, neuroprotective, anticonvulsant. Oleoyl-ethanolamine, it regulates food intake. Arachidonyl serine, it's vasoactive, acts in neuroprotective as well. And oleoyl serine was shown to have anti-osteoporotic effects. Now, there is limited information for the role of the endocannabinoid system during infection, especially against invading bacteria. And we wanted to see the role of the endocannabinoid system in against microbes. So together with Professor Doron Steinberg, we studied the antimicrobial potential of endocannabinoids and endocannabinoids-like compounds. Now, quorum sensing is a bacterial communication process. And bacteria, they use quorum sensing in order to attach themselves to surfaces and form bacterial biofilms. And the bacteria inside the bacterial biofilms, they are 10 to 1,000 times more resistant to antibiotics than the free planktonic bacteria. And we use here the Vibrio harvey bacteria in order to study the the quorum sensing, and because it emits bioluminescence as a result of, as a response to quorum sensing stimulation. And what we saw that the endocannabinoid anandamide, among other endocannabinoids and endocannabinoids-like compounds, it inhibited the quorum sensing of the Vibrio Harvey strains. It reduced the gene expression of the quorum sensing pathway, and it inhibited the motility of the Vibrio Harvey. Moreover, another kind of bacteria, which is the methicillin-resistant S. aureus, it is a kind of bacteria that is responsible for many difficult-to-treat infections in humans. And this kind of bacteria is a biofilm-forming microbe. And the biofilms, they are formed on the implanted medical devices, such as catheters, and on the necrotic tissues in wounds. And we were able to see as well that anandamide and the endocannabinoid-like compound arachidonyl serine, they were dose-dependently inhibited, showed inhibition for the biofilm formation. They inhibited the colony spread, and they reduced the bacterial aggregates. Addiction. Now, a publication in 2007 in Science stated that cigarette smokers with brain damage involving the insular cortex, they display cessation of tobacco smoking, which implicates that the insula is a region in the brain that may contribute to nicotine addiction. And therefore, together with another 3 groups in Canada, Richmond, Virginia, and Italy, we wanted to find molecules that were able to treat nicotine addiction. And we can do this by searching for neurochemicals in the insular cortex of the brain that can be altered by traumatic brain injury. And by using lipidomic analysis, we were able to see that by comparing traumatic brain injury and normal brain areas, that the traumatic brain injury elicited substantial increases in the endocannabinoid-like compound oledyl glycine. Now we wanted to see if the exogenous oledyl glycine will alter the withdrawal responses in the nicotine-dependent mice. The mice were given nicotine or saline for 14 days. Then the nicotinic group, they were given oledyl glycine or vehicle. 15 minutes later, they were given the nicotine acetylcholine antagonist mecamylamine, and 25 minutes later, they were evaluated by the elevated plus maze and physical somatic withdrawal signs. Now, the elevated plus maze, this is a test in order to assess the anxiety-like behavior in mice. If the mice, they are anxious as a result of withdrawal, then they will not be exploring the open arm. If they are not anxious, then they will be exploring the open arm. And what we saw is that the mice, the nicotine-dependent mice, undergoing mecamylamine precipitated withdrawal signs, they had reduced time exploring the open arm, and oledyl glycine actually increased the open arm time, indicating that it is attenuating the withdrawal symptoms in these dependent mice. In addition, this compound was able to attenuate or interfere with the physical somatic withdrawal signs. And these usually, they are seen as body tremors, head shakes, backings, curlings, and etc. In addition, we saw that oledyl glycine, it reduced the reward effect of nicotine. This was seen by a conditioned place preference test. The mice exhibited 2 neutral environments, and these two environments, distinct environments. They are paired spatially and temporally with unconditioned stimuli. For example, on days 2, 4, and 6, the mice were given drug in the dark compartment, and on days 3, 6, and 7, they were given saline in the white compartment, and on day 8, they are given the freedom to explore both compartments, and the time spent in any of these compartments is an indication of the reinforcing effect of the unconditioned stimuli. And what we saw is that oleal glycine dose-dependently inhibited the reward effect. It inhibited the development of the nicotine-induced conditioned place preference. Oleal glycine didn't interact with CB1 or CB2. However, it inhibited Pha, which is the enzyme responsible for the degradation of anandamide, and it behaved as a PPAR alpha receptor agonist. In order to see if we can do as well the same with morphine, we could find that oleal glycine, it blocked the aversive effect of the morphine withdrawal, and this indicated that oleal glycine might affect or decrease the impact of the morphine withdrawal, and this effect was reversed by a CB1 antagonist. In addition, there are somatic withdrawal signs for the morphine addiction, and these were seen as mouthing movement, abdominal contractions, diarrhea, and lying on belly. And what we saw is that oleal glycine as well attenuated these morphine withdrawal nausea somatic behaviors. And this effect, it was prevented by the PPAR alpha and CB1 antagonist. Now, oleal glycine, it has an amide bond, which means that it can be hydrolyzed in the body by amidases. So we thought of, is there any way of stabilizing this compound metabolically by introducing something to stabilize the compound. And in a similar molecule, which is anandamide, the introduction of a methyl group adjacent to the amide bond was able to potentiate its activity and stability of the anandamide, and therefore we prepared in the lab methylated oleal glycine and demethylated oleal glycine with the hope that methylation will do the same in oleal glycine as it did in anandamide. And indeed, we found that only the monomethyl oleal glycine, it produced a larger or longer lasting interference with the oleal glycine precipitated morphine withdrawal. And to explain this, if oleal glycine was given 10 minutes before the naloxone, then it had its effect. While if it was given 60 minutes before the naloxone, it didn't give any effect. But the monomethyl oleal glycine, 10 minutes or 60 minutes, if it was given before naloxone, then it gave its effect. And this effect was as well prevented by CB1 antagonist and PPAR alpha antagonist. And to summarize, we can say that the CBDA methyl ester is a potential medicine for treating nausea, anxiety, depression, and other disorders that are mediated by the serotonin 1A receptor activation. However, still we need more research to be done, more models, animal models, and etc. The endocannabinoid and endocannabinoid-like compounds, they may act as a natural line of defense against antibiotic-resistant bacteria. The endocannabinoid compound, like compound oleal glycine, it interferes with the nicotine withdrawal and with the naloxone precipitated morphine withdrawal. And a more stable compound, which is monomethyl oleal glycine, it may be more stable agent to combat the aversive effect of acute naloxone precipitated morphine withdrawal than oleal glycine. And finally, I would like to thank all of our collaborators, Roger Pertwee in Scotland, Linda Parker in Canada, Aaron Veller and Doron Steinberg in Israel, in USA, Aaron Lichtman, in Italy, Di Marzo, and thank you for your attention.
Hello, thank you for the great presentation. A question about these compounds getting into the brain if they're administered systemically or in the periphery. So what sort of evidence is there that endocannabinoid or endocannabinoid compounds administered in the periphery or systemically cross the blood-brain barrier and can affect the brain?
Well, we did some tests, and we have showed that, for example, when oleal glycine is given, and then it penetrates the brain. There is evidence, and there are studies, but I didn't show this.
Okay.
Yeah.
Would it also work for anandamide or 2-AG administered in the periphery to cross the blood-brain barrier?
Yes.
Okay.
Yeah.
Thank you.
You're welcome.
Hi there. I was wondering if you compared, when you were looking at the binding of the methyl ester CBDA to 5-HT1A, if you did compare it to the neutral form of CBD as well as CBDA or just to CBDA?
Actually, it is not binding to the serotonin 1A receptor.
Okay.
Yeah, it is activating it, and it was, yes, we compared it with the CBD and CBDA, and it was more effective than both of them.
Okay, cool, thank you.
Yeah.
Thank you for your overview. Quick question, is there any evidence for phytocannabinoid inhibition of your quorum sensing bacterial biology the same way you were talking about for the endogenous endocannabinoids?
Actually, in a previous paper, we did the same studies, quorum sensing and bacterial biofilm, with the HU210. Which is a synthetic compound that binds to both cannabinoid receptors, CB1 and CB2. And it did the same. In the literature, there is limited information regarding this kind of studies. There is only certain cannabinoids that are antibacterial. So, cannabinoids, antibacterial, 210, HU210, which binds to CB1 and CB2. It acts actually as anti-biofilm formation and anti-quorum sensing, and our studies on the endocannabinoids. This is the only data we have. Yes. Okay. Thank you.